Hello, everybody. In this lecture, we're going to talk about the intracellular structures of our eukaryotes. And we got a bit to cover, so let's get right into it. Again, like before, we have several objectives. In this case, you want to be able to discuss structure and function and location of ribosomes and microtubules slash cytoskeleton and the other components of the cytoskeleton. The structure and function of the nucleus, um, as well as the endoplasmic reticulum, Golgi apparatus, how vesicles and vacuoles work, and also the function of mitochondria and how they're similar to bacteria. All right, so we're going to start out with the ribosomes. They are going to be very similar to ribosomes in prokaryotes, and functionally they're the same. They make proteins, and they're made up of proteins and rRNA, or ribosomal RNA, which are a type of catalytic RNAs. The major difference is their sedimentation rates. So the ribosome for eukaryotes is an 80S ribosome, while their subunits are 60S and 40S. Now, when we talked about this S, that's a Svedberg unit. It's a measure of sedimentation, and that they're not additive. And so an 80S ribosome can be made up of a 60S and 40S. That doesn't equal a 100S. It just is how far it basically centrifuges out when you use an ultra centrifuge and you have it into a matrix that allows these things to separate. The ADS will centrifuge further down, so they're basically more dense. All right. Now, their primary goal is protein production, but they can be free or bound. And so that means they can be in the cytoplasm or they can be on the endoplasmic reticulum. And so the ones on the endoplasmic reticulum, which we're going to call the ER, produces proteins that are going to probably be secreted from the cell, while the free in the cytosol ribosomes are going to be used for making cytosolic proteins. Now, the localization of ribosomes can change between being free or bound, and depending on the needs of the cell. Now, here's the major difference is in the structure of the prokaryotic versus eukaryotic ribosome. You see that the sedimentation units, those Svedberg units, are different for the total ribosome, which is 70 versus 80, and for the subunits, the large being 50 in prokaryotes and 60 in eukaryotes, and the small being 30 versus 40. There's also some differences in the ribosomal RNAs, those ribozymes that are inside of the protein. They are similar, but they're not the same. And so this is another way they can be differentiated. Now, the cytoskeleton is going to shape the cell as well as play an important role in moving cargo throughout the cell. It's very responsive, and it's made of proteins, right? So the cytoskeleton is going to be made up of different types of proteins that make these filament structures. They move things from different places in the cell, and they help protect the cell from external forces. So this is exceptionally important when you think about eukaryotic cells that don't have a cell wall. They need something that protects them from those shear forces, those osmotic forces. The cytoskeleton can help with that. They direct the transport of vesicles. They help locate or move organelles around, and they're important for cell division. There are three main types. We have the microtubules, the intermediate filaments, and the microfilaments. And these are listed here actually from the largest, the microtubules, down to the smallest, the microfilaments. And so your microtubules are these hollow tubes. They're about 25 nanometers in diameter. They're made up of the protein tubulin. These are kind of like the roadways of the cell. So they're going to help move stuff throughout the cell. using these kinesin and dynein proteins as well. And of important note is that microtubules form the spindle during cell division, which is what helps you separate out your chromosomes. Now, these microtubules are going to arise out of these centrosomes. These are usually located near the nucleus. They're made up of two centrioles, and these are the same things that make up the basal bodies that anchor in eukaryotic flagella. Now, intermediate fibers are a little bit smaller. They're rope-like. Um, and they can be made up of a couple of different types of proteins, and they can have different arrangements, such as these monomers, dimers, tetramers, and here we have sheets of 8 or octamers. And these are important for protecting against that tensile, increasing tensile strength, protecting against, physical damage from the cell. Microfilaments are the smallest. These are very fine, 3 to 6 nanometers in diameter. They're made up these actin proteins, right? So here we have globulin actin, and then we have filamentous actin or G actin and F actin. This is the stuff that is associated with myosin, that's involved um, in muscle contractions. They help pinch the cells apart, and if a cell has a pseudopod, these can be important for moving the pseudopodia around. Here's the nucleus. Now, in the nucleus, it's going to have the DNA. It's going to be organized in the chromatin, and it floats around in the nucleoplasm. This is in contrast to inside the cell as a whole, which is the cytoplasm. And the nucleolus is this really dense area at the center of the nucleus. It's also enriched with RNA, and this is where the ribosomal subunits begin their development. Now, the nucleus is enclosed by a double membrane structure called the nuclear envelope, and they have holes in the envelope that are called nuclear pores. And this allows to control the traffic in and out of the nucleus. So things that need to get in or need to get out of the nucleus have to go through those nuclear pores. This is going to be continuously right up against your endoplasmic reticulum in certain places. So we can see here the nucleus, and down here we have our endoplasmic reticulum, or ER. The ER has an important role in lipid production. So in the smooth ER, it's going to make lipids. And many of the things that are made in the ER are going to get actually secreted out of the cell. Now, the rough ER is going to have ribosomes on it. It's going to be interconnected between multiple levels of these membranes, and they're going to kind of branch out from the nuclear envelope. Now, when we're talking about the rough ER, it's called rough because it's got the ribosomes on the outer surface. Proteins are directly inserted to the ER membrane, and they can be transported into the lumen. And you can make modifications to these proteins, such as clips and folds. You can add on organic and inorganic bits to your protein here as well. The smooth ER, on the other hand, does not have any ribosomes. You just have these kind of tubular looking bits that come off, and this is involved in lipid production, and it also plays an important role for detoxing the cell. The Golgi apparatus have like this function of receiving these vesicles that leave from the ER. So they bud off from the ER, and then they're going to carry something over to the Golgi apparatus. And so here we've got a transmission electron microscope image of the Golgi. You see these cisternae, which are these kind of like disc-like pardon me, disk-like sections, and they coordinate with the ER. They help modify proteins, build lipids, and they will help distribute things out to where they go. We got vesicles and vacuoles. Vesicles come in a couple of different flavors. They all have some sort of lipid bilayer. You've got transport vesicles, secretory vesicles, lysosomes, and peroxisomes. Transport vesicles are going to move substances around the cell, going along those microtubules. The secretory vesicles are going to move stuff outside of the cell, get rid of waste going along those microtubules. Lysosomes are going to have these hydrolytic enzymes that break down substances that were engulfed by the cell. All those things are going to move along the microtubules and the cytoskeleton. And peroxisomes contain enzymes that can break down molecules through oxidation, which produces oxygen radicals. And you got to keep those inside of these peroxisomes so they don't damage the cell. Now, vacuoles are like a conglomerate of several vesicles that merge together. They contain water and nutrients and sometimes toxins. They can be various sizes. They are found commonly in plants and fungi. And also they can be found in certain, in others as contractile vacuoles, which help regulate osmotic pressure that keep them from lysine in hypotonic environments. This is especially important for protists that don't have cell walls. Now, the mitochondria and chloroplast, they have these double membrane structures. This is where in the mitochondria, you're going to get ATP production. Chloroplast, you're going to do carbon fixation. They have 70S chromosomes that are 70S ribosomes and circular chromosomes. So those mitochondria are going to make ATP, amino acids, and vitamins. They regulate cell division. They take a role in apoptosis and cancer and some disease states. They have this double membrane, outer and inner. And then in that inner membrane, you got these cristae folds. And this kind of like, and then you got this matrix inside of that. It's kind of like a cytoplasm. Now, you can have different numbers of mitochondria depending on the type of cell you are, but almost all cells have mitochondria. Now, not all cells have chloroplasts, but plants and other photosynthetic organisms are going to have chloroplasts. But even if they have chloroplasts, they also have mitochondria. The chloroplast basically allows you to use to capture sunlight energy in these pigments like chlorophyll and convert that into sugar. They have inner and outer membranes. They've got these thylakoids that get stacked into these grana, and those are going to be important for the actual photosynthesis process. All right, and so with that, you should be able to describe the structure and function of these ribosomes, the cytoskeleton, the nucleus, the ER, the Golgi, the vesicles and vacuoles, the mitochondria, and the chloroplast. Where possible or where appropriate, you should also be able to compare that to how they work in prokaryotes. Well, I hope you enjoyed, and I'll see you soon.